On the inner membrane of the mitochondria lie a series of metabolic complexes collectively known as the electron transport chain. They help fulfill the essential final step of aerobic respiration, harvesting energy from NADH and FADH2 to ultimately power the synthesis of ATP. In the last few decades, considerable research has been done into the impacts of disruption to the electron transport chain, yielding some surprising findings. Induced mutations typically reduce an organism's fitness, which should come as no surprise. An average organism is generally going to be pretty well adapted, so randomly messing with its genome will very rarely be beneficial. But some surprising 1990s experiments, working with the model organism C. elegans, found several small mutations that actually increased lifespan. If these small changes were able to increase lifespan, why aren't they found in the wild-type genome? Well, we need to be careful not to conflate an organism's lifespan with its fitness. Natural selection favors fitness, which can be defined as the ability to produce viable offspring. Increasing the time over which an organism is capable of reproduction can increase fitness, but an increased lifespan with the same reproductive span would not. In fact, it would probably reduce it. If a sterile parent stuck around, it would only consume resources and provide competition to its offspring. In this case, a prolonged lifespan is disfavored by natural selection. Now that we have an explanation for why mutations can increase lifespan, we're left with one more question. How is lifespan extended? One promising theory held that these mutations were slowing down metabolism in the mutants. Metabolism has the unfortunate side effect of inducing oxidative stress through the production of harmful reactive oxygen species. So, if your metabolism is slowed down, you'll experience less oxidative stress and therefore live longer, right? This hypothesis was supported by the 1998 paper. For example, several of the mutations found to increase lifespan seemed to work by slowing down the cell cycle in larvae. Others were tied to the regulation of the entry of a dormant phase called Dower. But later work has called this theory into question. In the mid-2000s, the idea that impaired mitochondrial function was indirectly increasing lifespan by triggering some sort of compensatory mechanism that happened to increase lifespan as well gained many supporters. This has become a leading theory in explaining the increased lifespan of the mutants, as the original rate of living theory has been put into serious doubt by research since then, including the findings that increased ROS exposure was not directly negatively correlated with lifespan in C. elegans, and that exposure to ROS could actually increase lifespan in some cases, suggesting that oxidative stress may trigger the mechanism that increases lifespan. Regardless, I wouldn't completely write off either theory. A variety of mutations have been found that increase lifespan in C. elegans, and they may be acting through different pathways. And I wouldn't even be surprised if a new theory is proposed in the near future that even better explains this counterintuitive phenomenon. Talk of increased lifespan may get you excited, and with good reason. Are these findings at all applicable in humans? I don't really think so. This body of research has helped us understand a lot of human mitochondrial diseases better, but I'd say we're too dissimilar from nematodes like C. elegans to be hopeful that this line of inquiry lead to a magical increase in human life.